So what we're going to do is we're going to start up. So this has all been done in Binder, which is a fun little way of making sure that everybody has the tools that we want access to. You can launch the Binder by clicking this button here. It might take a second for Binder to load up. Sometimes it takes a while, depends on the last time it started. And it also, one of the things to know is that there are 3 different areas for Binder. So when you start up a Binder window, it is open and the little instance that you're generating is open for 6 hours. If I open one and then somebody else comes and opens up the same Binder within that 6-hour window, it generally opens up faster than having to build it from scratch. But one of the downsides to that is that there are 3 mirrors for the Binder interface. And so if I do it here in the US and you're in either Asia or Europe, and then go to try and launch the Binder, it might also take some time there, even if everybody in the US is able to hop on really quickly because it's been launched recently. What we're gonna be able to do here is we're gonna process through some of the data to allow us to look at how Prodigal runs. And so if you're looking at the screen right now, so we'll come back to this in just a second. So this is Binder. Binder has launched. We're going to launch from here. In the repo, the GitHub repo for this particular lesson, you can go into lesson 1 and it highlights the walkthrough contents here. So this is what we're going to mainly be doing. Like I said, it's going to be relatively quick. And it's going to give us an idea of how Prodigal works. We're going to look at some of the outputs from Prodigal as part of the tutorial. And then we'll move on from there and open it up to questions. And if there are questions as we go, again, feel free to raise your hand and we can answer them. Okay, so we're back here. We're in our Binder. If you are still loading it up, I'll give it another second. But what we're going to be doing is we're going to be clicking on this button here that says Terminal. And that's going to generate a terminal window for us that we'll then be able to work around with, just like it was any other Unix environment that we're playing around with on our computers or on a server or something like that. So I'm going to click it now. All right, and so this is a Unix instance of the repo, the GitHub repo that we started in, right? So you can see here on the left, we have our directories, which we'll slowly fill out with other lessons. Environment.yml that generates this Binder environment and told it to install Prodigal for us. The README is that main README on the main page. And so we can do very normal Unix and Linux based terminal things here. So first thing we're going to do is we're just going to run ls to list our environment. It should hopefully very much so mirror our components that we have over here. And then we can move into the lesson 1 directory using the cd command. And here, if I hit ls, you'll see that we've changed and we're now in have different files available to us, including a FASTA file from a metagenome assembled genome from the Poseidon-Alles order. And so the tool that we're interested in the most is Prodigal. So what we're going to do is we're going to run Prodigal with its help menu displayed and chat about a little bit of what types of flags that we're going to use to make sure that we're getting the output that we want out. So Prodigal-H produces this output for us. And then let's just walk through this. So the -A is going to be the file that we want to put our protein sequences in. Right? Seems pretty basic. The default output of Prodigal is not a FASTA file with proteins. Our default output is a GFF type file, which we'll look at in a second, which you can then convert back to a FASTA file, but it's much easier to just have it output your FASTA file to you. So that's what the -A does. -C, so this you would use if you're using a circular genome. The idea here is that Prodigal is expecting your genome to come in multiple fragments. And so in that instance, it's able to look at a start codon, get to the end of your gene, your sequence fragment and say, you know, this is the end of the fragment. I had a start codon. It looked like a protein. let me give them half the protein back as opposed to saying there's no protein here. You can do it also on the starting part of the fragment as well. So if you had a stop codon and it looked like a real protein up to that stop codon, but no start codon at the start of a fragment, it can do that. And so saying that it's closed will tell Prodigal to make sure that like, don't look for anything that looks like that. You know, everything should have a start and a stop because we're dealing with a closed program or closed genome. Don't assume otherwise. The -D will write out the nucleotide sequences of the genes that you're working with. So it's a partner to the protein translations. So you'll get the open reading frame in nucleotide space and then the converted protein. You can select an output format that is different than the default one, which is a GenBank format. You can specify the translation table, which is set at 11, which is the standard one for bacteria and archaea. This is the help menu. We need to tell it what FASTA file it's reading in. And so then this one is really important when you're dealing with assemblies, this -M, which says treat runs of ends as masked sequences and do not build genes across them. So when we're dealing with environmental sequences and we deal with environmental assemblies, there's the possibility that the assembler can't actually discern what the correct base pair is at a position. And so one N isn't a problem, but multiple N's in a row might actually represent, instead of a true length of a sequence, it might represent a gap where we actually don't have any information. So instead of trying to build a protein across that gap, which might result in a chimera, Prodigal just says, no, I'm not going to do that. Bypass the Shine Dalgano trainer and force a full motif scan. So this is specifically the motifs that are being looked for as part of the ribosomal binding site upstream of your start codon. We can specify the name of the output file as opposed to its default, which is written to the screen. We can select the procedure, the difference between single and meta, which we'll talk about in our example. We can tell it to run quietly, which we won't. We can then also ask it to write out all potential genes. So like I mentioned in the lesson and in GeneMark S2 as well here in Prodigal, Prodigal does this part where it looks at all the open reading frames and their orientation, and they might be overlapping, and selects the best one. This would allow you to look at all of those options as presented to you from Prodigal, along with their scores if they exist. So the other -T we can use to write out the training file. And so we'll talk about this again when it comes to single and meta in just a minute. And then the -V, which gives us the version of Prodigal we're working with, and then nothing else. So what we want to do is we want to take our starting FASTA file, this med921 Poseidon-Alias FASTA, and we want to output proteins and genes that are associated with those proteins. So let's put a little bit of text on the screen and write that out. So we're going to do Prodigal. So we're going to give it our input, which is our med921. At this point, this Unix command line functions like any other command line, so I can actually hit tab to complete the rest of this file name. We're then going to write out the proteins to a file. And so I'm going to call it med291. We're going to call it proteins. We're going to call it single. That's what we're going to run. And we're going to say FAA. And so FAA is in a different abbreviation. So we're starting with a FASTA file. This is also a FASTA file, but it's a FASTA file with amino acids. Look at that. Look how easy that is to it's like somebody thought about it, like really hard. We're going to do this again, but instead of proteins, we're going to call these genes. Also going to run it with single. And then here we're going to call it FNA. So it's a FASTA with nucleic acids in it. At that point, we only have a couple more flags. We're going to say don't. We're going to use this -M to treat the runs of ends as a masked sequence and not to build information over that. We're going to use this, instead of printing the standard out to the screen, we're going to put it in a file, which we're going to call Prodigal med921 -single -out. And this might be a little different than the example in the in the repository, but it's still going to work. And the last thing we're going to run and set, just -p, and we're going to say single. Okay, and so I've been mentioning that we're going to talk about single and meta, and this would be the perfect time to do that. So the difference between single and meta is that when we run it with a -p single, Prodigal uses its unsupervised algorithm to create the proteins that we want in this genome from scratch. Right? It goes in, it tries to learn what the average distances between ribosome binding sites is and the start of a start codon. It looks for all these orientation components and creates a model of the genome that we're giving it so that it's custom to the genome that was input. And so that's where this -t comes in. So if we wanted to preserve that model and use it, currently you'll see that this runs very quickly. But if we were doing this on something that was maybe larger or had we wanted to like really speed up the process where we didn't have to do this initial training step, you can actually import that training file and have that information ready to go. So once this is all typed out, this should work. If there's a typo, we'll correct it. And we can hit enter. So here's that training step. So it's looking for the different biases that might be custom to this one particular organism. It runs through all 35 of the contigs that are part of the genome and then finishes. Very fast. I think that was what, like 4 seconds, right? And if we hit LS, we have all of our single, we have our gene single, we have our protein single, we have our single output right here. And so we can do the exact same thing, but this time we can do it with the meta flag. And we're going to do this just so we can explore how these might be different. So we're going to put our same input. It's med29, Poseidonales. We're going to remind myself A and D. That's what we want. Med29. And this time we're going to call it proteins-meta because that's the one we're going to run it with. We're going to do the same thing with the genes, which will be -meta, -FNA. And then we're going to say -m to make sure that we don't go over the runs. Yep. We're going to do a -o where we're going to have our Prodigal output. And then say meta.out. And then again, we're going to do this -p. And instead of single, we're going to write meta. And I'll give everybody a second for that. And so what's going to happen here is as opposed to training the model on the genome that we're submitting, Prodigal is going to use the existing models that it has to predict which model best reflects the genome that we're looking at. And so we're going to run that now. Again, this should run fast, probably even faster than the last one, maybe a little slower. Okay. You do you, Prodigal, that's fine. And so then what we're going to do when this finishes up is that we'll look at the various outputs that we've generated, and then we'll do a really quick comparison. And by quick, I mean, we'll look at the numbers as opposed to looking at any of the specifics as to what it is. The specifics will come up later in the lessons as we go forward. Great, so this is done. So we can hit LS. So we generated 3 types of files, right? We generated our protein file, we generated our gene file, and we generated the Prodigal output file. So let's just take a look. We can take a look very quickly at either the meta or single version of these. We're going to use less to take a look at it. I'm going to do med 921 proteins. Let's pick meta for right now, and let's bring this up. So this looks like our own standard, a regular protein file in FASTA format. So we have our leading caret to denote our header lines. We have an ID. So all Prodigal has done is taken the contig name, which is this first component here, and then appended a number and then added it sequentially. So this is gene 1 on contig 1, gene 2 on contig 2, gene 3 on contig, et cetera. Prodigal does not promise, because you're the one submitting this, does not promise that this header right here, this identifier is unique. it does promise within a file that this identifier is unique. So this also mirrors that count up, right? So this 1-1_1_1_2 mirrors the 1 and 2 here. And then you can see that there are these pound signs. So these pound signs are denoting the next sets of information. This is your start position on the contig that you are working with. So it's the 104th base pair. The protein goes to the 2725th base pair. The -1 here says it's on the reverse strand as opposed to a +1, which is the forward strand. And then we get into information that's very specific to the prodigal output, which are separated by these semicolons. So this was the ID. Here, partial is represented by 2 zeros. The zeros indicate, so it's a binary system of 1 or 0. If you have 2 zeros, it means that the protein that you're looking at, the gene that you're looking at, had a start and a stop codon that was real. If there was a 1 in this position, it would mean that this protein was missing its start codon. And if there was a 1 in the second position, it would mean that it's missing the stop codon. And so that would give us information. If this was something that was at the edge of a contig that we were looking at, you would maybe not be surprised because Prodigal can account for that. Because if it was happening somewhere in the middle of the contig that you were looking at, it might be because you have that run of ends that we were talking about with a bad assembly. And it would give you an idea of how confident Prodigal was that there was this looks like a true gene here. It tells you what your start codon was. It tells you if there was a ribosomal binding motif present, which it says there wasn't. How long that spacer region would have been if there was a ribosomal binding spacer region between it and the start codon, and then the GC content of the gene. And so if we look at, so that was our proteins. If we do that again for our genes, what we'll see is 1, we're in nucleotide space now. 2, that the header is exactly identical to that first one that we saw in the proteins, right? So they're exactly mirrors of each other. They maintain that identifier information, and they maintain the same header information. We can also look at the Prodigal output, the raw output that it would get. I'm gonna look at the meta one here. So this is a GFF file format, and there's a lot more information here than on the standard output. Some of the things I'd like to point out is it stores which version you're running of Prodigal. So if you have this file and you're writing your manuscript and you have your methodology, you can track which version you used. It tells you which type of run it was. So this was the meta model, right? So metagenomic. And then here it tells you specifically which model it used based off of what it thought was the right way to address your sequence. And so in this instance, it's Haloquadratum Walsbyi, which is an archaea. The genome that we ran was an archaea. And so that is good that we have that information. It tells us our translation table. And then it gets into specifics about the confidence and the scores that are how Prodigal is deciding that this is a good or potentially good sequence to work with. Okay, and so I want to show you really briefly the same thing from the single output. The single, right, so we ran in single type mode. The model that was then predicted was ab initio, right, so that it didn't use one of its predetermined models already. And so when we actually look at the output from this, if we did very quickly proteins, and instead of meta, we did single, we still get very similar information here, right? Again, that this is a partial, that this is a full, has a start and a stop codon, that what the start codon was, that there wasn't a ribosomal binding motif, and there weren't ribosomal spacers for this particular organism. When we get further down, you can actually see that there is information about the ribosomal binding motif, right? So here's what the binding motif looks like, the family that it belongs to, and then how far upstream it was of that start codon. So this would have been 5 to 10 base pairs upstream of the start codon, and that's throughout the rest of the file. All right, I said I wanted to talk about how these outputs compare. So one of the ways that we can do that in a really kind of brute force method is to look at how many proteins are actually predicted for this particular for these outputs. So we're going to use that using the Unix command grep, right? And so we're going to use grep. It's really important here to make sure that you use the quotes here. We're going to grab the header line from the file. So this will give us a count of how many unique headers there are in any one file that is in FASTA format. We can do that with proteins, meta. So you can do this with the genes, you can do it with the proteins. And then we're going to pipe it to the Unix command word count. And we're just going to ask it for how many lines are present here. So this will just tell us how many times did it find a header, and each of those headers represent a unique gene. So I'll hit enter here. The number is 1,389. That seems pretty reasonable for a microbial genome, especially a microbial genome from the ocean where it is coming from. And then we can hit the up button to recreate the last command. And then we can edit it and type in single and run this to see if there's any differences, which there are. There are 4 genes different between the meta prediction and the single prediction here. Like I said, we're not going to get into the details about what those 4 are. We'll get into that. We'll use this as a way to continue going forward in the other lessons to look for and how we would determine if those 4 genes are important, but there are different outputs there. One of the things that is also interesting to do is, like we've seen a couple times now, if I just show you this again really quickly, there's a lot of information that is present here on this header line. And a lot of programs don't have a problem with this. Some of them might. There's a pretty quick way of reducing that information to just the identifier. And we can use that using the Unix command cut. So using cut -F1, the standard component that is used here for cut is a tab. I'm pretty sure, I'm doing this on the fly. This is not part of the written lesson, that there's a space there. So we're gonna need to use a -D and then put a space between quotes. We're gonna target this particular protein, and then we're just gonna put it into a temp file that we're gonna call temp.fa. And so now when I look at that file, all of that information has been removed here, but has been preserved. The rest of the sequence has been preserved here. And the reason for that is that cut is looking for that first space after the start of the line. So in the header line, you have your identifier, which comes with the caret, which is part of the format, a space, which then denotes the rest of the information, which we don't want to keep. And then each of the lines after that is just that one line. It's 80 characters of either your nucleotides or your amino acids. So cut knows that that's what you want to keep. So we can do that and generate a FASTA file, either of the proteins or of the genes, that then have a reduced header information for going forward. But that's just a little thing that you can do to clean things up a little bit. But with that, that's the end of the tutorial. Thanks for tuning in, and we'll be back with lesson 2.